Hello, my name is Cayah Miller, and I am a senior biology major on the pre-med track here at Spelman College. My journey as a member of the Ethel Waddel Githii Honors Program has been one of growth, exploration, and connection. This program did more than challenge me academically. It offered me a community of curious, creative, and committed thinkers. It helped shape my identity as a future physician scientist who understands the value of interdisciplinary knowledge and leadership rooted in purpose. Today, I'm excited to reflect on 5 portfolio artifacts that highlight how my intellectual and personal identity evolved during my time in honors. Each piece represents a unique chapter in that journey, whether in the lab, the classroom, or how I've come to view my role in advancing equity in science and healthcare. The 5 artifacts I will be going over in this video are Microbiology Unknown Bacteria Lab Research and Report, PUMA Summer Internship Research and Presentation, Developmental Biology Arthritis Research Paper and Presentation, Colorectal Cancer Research Project and Presentation, and Lactase Persistence Gene Research Project and Presentation. Artifact 1, Microbiology Unknown Bacteria Lab Report and Research. The first artifact I'm sharing is my Microbiology Unknown Bacteria Identification Lab Report, a project that began with a simple swab of the mouth and grew into a deep investigation into the world of oral microbiomes. What seemed like just another lab assignment quickly became a pivotal experience for me. I wasn't just following steps, I was thinking like a microbiologist. Throughout the process, my partner and I performed a variety of tests: gram staining, catalase and oxidase assays, starch and gelatin hydrolysis, and EM blue plating. These techniques helped us gradually eliminate possible bacterial identities until we concluded that our sample most closely resembled Neisseria, a genus of the commensal bacteria found in the mouth and nose. What made this project special was that it taught me the values of scientific troubleshooting. We faced contamination issues and variable growth rates, yet we adapted. We had to interpret results not just for what they were, but for what they could mean given the limitations of the lab. The Githii Honors Program had already introduced me to the importance of inquiry and reflection, and this experience showed me how those values show up in the lab as much as in writing or in dialogue. This artifact helped me strengthen my lab technique, sharpened my scientific reasoning, and reminded me that science is not about perfection, it's about curiosity and persistence. Artifact 2, PUMA's Summer Internship Research and Presentation. My second artifact is my PUMAS research presentation from my summer at Gladstone Institutes in San Francisco, California. I was fortunate enough to work in the Rowan and Ott labs, researching how COVID 19, especially long COVID, affects the brain using brain organoids and iPSC derived microglia. We used human stem cells to grow dorsal forebrain organoids, essentially mini brains in a dish, and paired them with microglia, the immune cells of the brain. While the project was in its early stages, we laid foundational work by growing and differentiating these cells, learning to visualize their structures under a microscope, and outlining future directions for gene inhibition and viral exposure testing. This project wasn't just a research opportunity, it was transformational. I stepped into a lab full of postdocs and graduate students, and instead of being intimidated, I felt empowered. Honors gave me the confidence to ask questions, propose ideas, and see myself as a part of a team working on real world scientific problems. What I appreciated most about this experience was the way it connected academic interest to a larger societal issue. The neurological impacts of COVID 19 disproportionately affect underserved populations, and our research could eventually inform treatment strategies. This work planted a seed for my future in reproductive endocrinology, where I hope to use stem cell technologies to treat hormone-related reproductive conditions. My time in the Githii Honors Program helped me build this scientific and ethical grounding to approach this kind of research with both rigor and responsibility. Artifact 3, Developmental Biology Arthritis Research Project and Paper. The third artifact is a term paper I wrote titled A New Dawn in Rheumatoid Arthritis Care: The Stem Cell Revolution. This project was incredibly personal. I structured the paper around a fictional character named Teresa, a middle aged woman navigating the emotional and physical toll of RA. Through her story, I examined a range of clinical trials involving mesenchymal stem cells, or MSCs, and other cell based therapies that show promise for long term RA management. This was the first time I truly fused science with narrative storytelling, and I found it to be a powerful medium. The paper wasn't just about listing study results, it was about humanizing the science, making it accessible to people like Teresa who are actively seeking better treatment options. This project helped me realize how passionate I am about science communication. Honors has always encouraged me to find my voice, and this paper is where I discovered how to use that voice to translate research into hope. I dug into experimental designs, patient outcomes, immunomodulatory mechanisms, and ethical considerations. But what I'm most proud of is how this paper seeks, speaks to real people facing real challenges. It also aligned beautifully with my interest in regenerative medicine and personalized care. I envision a career where I not only conduct and apply this kind of research, but also advocate for patients' understanding of their options, especially in historically underrepresented communities. Artifact 4, Colorectal Cancer Research Project. My fourth artifact is a group presentation on MSH2 mutations and their role in hereditary non-polyposis colorectal cancer, or HNPCC. This was one of the most advanced and technical lab projects I've worked on. It involved a multi step process using plasmid digestion, PCR amplification, yeast transformation, and FOA assays to test how different plasmid mutations affected mismatch repair function. We analyzed three unknown samples and identified which one contained the mutated PMSH2 gene based on protein protein interactions and gel electrophoresis results. This hands on experience brought molecular biology to life in a new way. Suddenly, all the theory I'd learned about gene repair and cancer susceptibility had a tangible, testable presence in front of me. What made this project meaningful in the context of honors was how deeply it connected to public health. HNPCC is a hereditary condition, and learning how to detect these mutations at a cellular level deepened my interest in genetic counseling and the future of personalized medicine. It also gave me the chance to improve as a team researcher, balancing technical precision with collaborative communication. Honors has taught me that true scholarship isn't done in isolation. It's born out of rigorous conversation, shared curiosity, and a willingness to learn from others. This presentation was a great example of all of that in action. Artifact 5, Lactase Persistence Gene Research and Presentation. The fifth and final artifact in my portfolio is my Evolution in Action project on lactase persistence and geographical variation. For this, we analyzed LCT gene sequences from African, Asian, Middle Eastern, and European populations to trace how small genetic mutations shaped humanity's ability to digest dairy. Using FASTA alignments in a phylogenetic tree, we demonstrated how missense mutations in the LCT and MCM6 genes reflect patterns of gene culture coevolution. Populations with histories of pastoralism, for instance, are more likely to carry genetic traits that allow lactose digestion into adulthood. What made this project impactful for me was its intersection of science, culture, and evolution. It was an elegant example of how our environment, behaviors, and biology are interconnected. I was able to see evolutionary theory play out in human genetics, and I could also see its implications for healthcare today. From dietary recommendations to global health initiatives, understanding gene culture coevolution is key to reducing health disparities. This project reminded me that as a scientist and future physician, I must stay attuned to the diverse lived experiences that shape our biology. The honors program has always taught me to value multiplicity, to embrace the complexity of identity, history, and heritage in every intellectual pursuit. As I reflect on these five artifacts, I'm reminded of just how far I've come. From navigating lab techniques and writing research narratives to investigating evolutionary questions and studying genetic diseases, I've developed as both a student and a scholar. But the most meaningful growth has been internal. The Ethel Waddel Githii Honors Program didn't just challenge me to achieve, it taught me to lead, to serve, and to think expansively. It gave me a sisterhood of brilliant Black women who pushed me to stay grounded in my purpose and bold in my ambitions. Every artifact in this portfolio represents a moment of learning, resilience, and transformation. Each one prepared me to step into the next chapter of my journey, not just with technical knowledge, but with a sense of direction and responsibility. Thank you to the honors program, to my mentors, and to the community that has nurtured my growth. I carry this experience forward into medical school and life, where I hope to continue bridging research and care, science and storytelling, and intellect and empathy. Disclaimer: the Canva link will be linked below in the description because there are interactive parts of the presentation that show my artifacts that could not be viewed during this recording. Thank you.